We have our representatives from our land conservation offices in each of the 3 counties that were involved in the study. I'm going to start with Erica Sauer, who is department manager. If you could raise your hand, Erica, with Lafayette County Land Conservation Planning and Zoning. We also have Katie Abbott, the county conservationist with Iowa County Land Conservation Department. Eric Hagel, relatively new administrator for Grant County Conservation, Sanitation, and Zoning Department. And we also have with us Linda Schweikert, who was the former administrator for Grant County Conservation, Sanitation, and Zoning. We invited Linda to come here because Linda was very instrumental in getting this study done and getting it underway. So we wanted to have Linda back here and recognize her for her involvement in that. So I'm going to go over to our researchers. We have Joel Stodick, who is a biologist with the U.S. Geological Survey, Laboratory for Infectious Disease and the Environment. Mark Borchert is Emeritus Research Microbiologist with USDA Ag Research Service. Maureen Muldoon is a groundwater researcher with the Wisconsin Geological and Natural History Survey with UW-Madison Division of Extension. And Ken Bradbury, there's Ken, who is also with the Geological and Natural History Survey. He is the director and state geologist. And without further ado then, I think I'm going to turn it over to you, Mark.
Is this on okay? Great. So my name is Mark Borchert, Emeritus. That's a fancy way of saying I've been retired for 1 week. So it's my first week in 53 years without a paycheck. It feels really weird. I've got 42 years of research experience. I've got 30 years of research on groundwater and microbiology. I'm one of the 5 researchers on this project. The reason I'm involved is the SWIGG study is modeled after a study for which I was the principal investigator, the Kewanee groundwater study, which you may have heard about. Tonight's presenters, there's 5 members of the research team. First of all, it's me, and I'm going to talk about the study background, and I'm going to give you a little information on the role that we have as scientists. After that is going to be Maureen Muldoon speaking about the hydrogeology of where you live, what's underground, what's important for your groundwater. Then we get to the heart of the report, and that's Joel Stodick in my group. So I'm the program director. Well, I was last week, not anymore, but at the Laboratory for Infectious Disease and the Environment. And Joel is going to spend a lot of time talking about the groundwater contamination and the risk factors for that. And then Ken Bradbury is the Wisconsin State Geologist, and he's going to talk about the sorts of things you can do to protect your private well. Those are the presenters for tonight. And then I listed Bernie Kiki. He's a biostatistician at the Marshfield Clinic, and he's a person I've collaborated with over the last 20 years, and he's responsible for the statistical models, of which Joel's going to show you some of those. This is a large study, therefore a large study team. We could not have done this study without the people over here, the county conservationists. You can't do a study of this scope or with this hands-on, boots on the ground work unless you have buy-in from the counties. So we'd like to thank Linda, Katie, Terry, Eric, and Erica. Laboratory and field sampling was done by my team, of which Sue, also retired now, and then Aaron, still back in Marshfield. We're located in Marshfield, Wisconsin. And then we thank Scott Laser and Clean Wisconsin for their support and their perspective on the issues in the 3 counties. Continuing on, there's a lot of people we have to thank for the data that they provided us with. Some of these things in blue that are underlined, well geolocating, geographic information systems, septic and sludge records, and precipitation, that will all make sense when Joel speaks. But I have to thank the people that helped us get this data that allowed us to do this statistical modeling. And then funding. I've never been part of a study that's had so many funding sources. The counties themselves, 3 levels, 3 governmental levels, from county, state, to federal funding. And impressively, residents of Lafayette County that also donated to this cause. And then the U.S. Geological Survey, my group, the USDA Agricultural Research Service, and the Wisconsin Geological and Natural History Survey. All these funding agencies came together to support this study. So how did this all start? I blame Linda. Linda gave me a call, and she asked Madeline Gotkowitz, who at that time was a hydrogeologist for the Wisconsin Geological Survey, and myself to speak to the Grant County Board. At that time, Linda can give more background information. There was interest in a moratorium on manure spreading and some restrictions on that. So I spoke about the work that we were doing in Kewaunee County. Madeline has a long history of research on the hydrogeology of this area. We spoke, and Linda invited Iowa and Lafayette counties. Grant County decided to do the study, and Iowa and Lafayette counties joined in. Get my slides back. To be honest, I wasn't too keen on doing this work. And the reason is that I was in the thick of Kewaunee County and everything happening there. And also scientifically, I just didn't know what was going to be added. But Madeline Gotkowitz convinced me that your hydrogeology is unique and would be of scientific interest. And you're going to see that tonight when Dr. Muldoon speaks. One more. And then the next one. Thanks, Eric. To give you a little background on the research process, how this worked for us, is that we started off talking to the counties, what their questions were. We developed study objectives. We develop a study design to meet those objectives. That is, we come up with the methods, the approaches to answer the questions that the counties had. We wrote a research proposal, and that was reviewed by the counties and decided to fund it. We launched the study, we enrolled participants, we sampled wells, we did the lab analyses, we did the statistical analyses, and where we are right now tonight is here. We are here. We're presenting the report, which will be available online at this moment. So that's where we are, and we've met the obligations with the counties, we've done the work, and we're reporting the data. In addition, so that you are aware, there's one more step for us, and that is we write the scientific papers, because that's our currency. That's how we communicate with other scientists around the world. So that's going to be happening this fall, and our goal, Joel's and my goal with that paper, is to get that sent in and submitted, hopefully published by maybe January or February of next year. This is an example of the sorts of things that we do. These are the two papers that were published on Kewaunee County, and what we strive to do. If you're interested, these are free. They're available online. If you want to see the next step that we're going to be taking and what we're going to be doing with the SWIGG study, it's going to be something along these lines. For this study, both studies, Kewaunee and SWIGG, they were designed to achieve two goals. State-of-the-art assessment of private well contamination. We use statistical methods and sampling programs or protocols for getting state-of-the-art knowledge. But that's not enough. And one of the things that I was told when I joined USDA Agricultural Research Service 12 years ago was it wasn't enough just to point at problems. We have to identify solutions too. And that was the second goal, to come up with findings that help address these contamination issues and give policymakers the tools that they need to make decisions about what could be done to improve the situation. Those were the two major goals of these two studies, and I think we achieved them with the SWIGG study here you'll see tonight. What's our role as scientists? And I want to be very clear about this, so you understand what we do and what we don't do. So we apply our skills as scientists to design the study to meet specific objectives and questions. We don't head off this way or we head off that way. We have a goal in mind and we want to meet that goal. We apply our appropriate technology, some of which Joel's going to talk about tonight, to meet those objectives. As much as humanly possible, and I know this is a strange thing to say nowadays, but we strive for objectivity and we strive for transparency. Nothing is hidden in what we do. I can't say all scientists behave that way, but it's what we strive for in our group. And what we do is we provide and interpret the scientific results. We do not make policy recommendations or management recommendations, and we do not advocate for a certain point of view. We don't do that. But as I told some of the county conservationists earlier tonight, we are happy to come back and help provide further interpretations of the data, help answer questions, and continue to help these 3 counties. Why do we do this? Well, this is a little play on the words of think globally, act locally. Instead, what we like to do is we research locally, publish globally. So the benefit of what we've done, and you're going to hear about today, is that we do local research that has scientific knowledge that's of benefit to the residents of the 3 counties. So it's information that's specific to where you live. But at the same time, when we publish these findings in scientific literature, this goes worldwide. So the Southwest Wisconsin data will be published worldwide, and it advances groundwater science, and it can be used by other scientists and other people to aid their groundwater quality issues. You need to understand, and I'm going to brag a little bit here, this study, the SWIGG study, is only the second time, only after the Kewaunee County study, only the second time in the scientific literature that we've related microbial contamination, that is the microorganisms that are contaminating groundwater, to risk factors, those sorts of things that lead to contamination. That has not been done before, other than Kewaunee County. So you're only the second place in the world that this has been done. And then lastly, the way I want to end is that when I speak at scientific conferences, I have a number of people come up to me and say, how do you do these private well studies? How does that work? Because they're private wells, they're privately owned, it's on private property. How do you get access? And what I say is, well, we work in Wisconsin. People are nice, people are neighborly, people are concerned about the environment. So people are always amazed by the number of people that we have to participate. In this study, 816 households volunteered to participate. And we cannot do work like that unless people volunteer. So for those of you in the audience that allowed us to sample your wells, thank you. Thank you. You really contributed to what I think is a major scientific study. And with that, I'm going to pass the microphone on to Dr. Muldoon, and she'll speak about the hydrogeology of your region.
Hi, so I am the groundwater scientist on this study. And so what I'm going to do today, my major role in this project was to try to get a better understanding of how your groundwater flow systems worked in this part of the state. So what I'm going to do is first I'm going to talk a little bit about the geology of the area, both the sediments on top and the bedrock, and then I'm going to talk a little introduction to how groundwater works, and then the specifics of the groundwater flow systems. So I'm sure you all know that you guys live in the Wisconsin's driftless area. On the left here, we have a map of where the glaciers were at the last glacial advance into the state of Wisconsin. And you can see that this southwest part of the state was not covered. And this is a figure from Eric Carson, who's one of my colleagues at the survey. He's done a lot of work in the driftless area. And this kind of yellow outline here is the true driftless area where the ice never came. This other line shows just kind of areas where the topography is a little different. But I live in Oshkosh, where it's really, really flat. Right? And one of the things about the driftless area is you have a lot more hills, and then you have those deeply dissected streams. So the topography in this part of the state just doesn't look like other parts of Wisconsin, certainly not like Oshkosh. Okay, I'm sure everybody in here has probably heard the word karst, but I just want to take a little minute and sort of explain what karst is and sort of where we expect to find it in Wisconsin. So karst is a landscape. It's not a single sinkhole or a single cave, but it's a whole landscape where the bedrock, typically either dolomite or limestone, slowly but steadily dissolves as water flows through it. And that's how we get caves, that's how we get sinkholes, that's how we get conduits. Right? So this is a figure that I took from Minnesota Geological Survey, and you can see a lot of the features of karst. So there's little sinkholes, there's sinking streams, there's caves, and here's a map of where we have these features in Wisconsin. So typically what you need is sediments that are 50 feet or less in thickness over that dolomite or limestone bedrock. And that's this U-shaped pattern around Wisconsin. And as Mark mentioned, we both worked on the Kewaunee project. And over here, you have one type of dolomite. But when you're forming the karst over here, you're actually in a different geologic setting where you have dolomite, but you also have things like clays and shales and sandstones. And that's going to come up more later, partly in my talk and partly in Ken's talk, about how that affects your groundwater flow patterns and how that affects your geology. Okay, so karst features, just a couple pictures. This is from the survey. This is a sinkhole. This is from Department of Transportation, just a little conduit in a roadway. You guys live here. You've seen stuff like this. I want to talk a little bit about the superficial geology. So when you take a dolomite or limestone and you weather it, sometimes you get some residual material. And sometimes it's kind of clayey, sometimes it's other, maybe a little sandier. But the unit down here is called the Roundtree Formation. And like I said, it's a weathering product, which the fancy name for that is residuum. And that covers part of the landscape here, and we're going to look at it a little more in the next slide. But here's a picture of one of my colleagues, Grace, taken by Eric Carson. And what we have here is we have that residual, very red, soily material, sediments. And on top of it, we have loess. And loess is actually, it comes during the glacial time period. It's sort of silt-sized sediments that are, as water melts off the glacier and deposits a ton of sediments, the wind picks it up and blows it across the landscape. So typically, it's the top unit in your surface geology. So you have loess with Roundtree underneath. And this is a map being made by Eric Carson. He's got Grant done in Iowa. He's still working on Lafayette. But this is showing basically where the Roundtree is and where it isn't. And these units are not coming through great on the slide, but this kind of tanny color, those are in stream valleys. So you either have slope wash on the sides of the stream or you have sand and gravel in the bottom. Okay, so that's sort of what the tan stuff is. This, oopsie, I was trying to hit the pointer. This kind of pinky salmony color, which is through here and here, that is basically where you just have loess. You have no Roundtree. Okay, so it's just that silt-sized sediment. And if I actually check my notes, because Eric gave this to me pretty recently and I don't have it memorized, in Grant County, it's about 27.5% of the county that has Roundtree. In Iowa, it's about 17.8%. So it's very patchy in its distribution. So this pink stuff is just plain loess. This kind of darky brown color is where you have loess over Roundtree. So you can see it's not really everywhere. And this becomes important when we think about groundwater because people thought, oh, that clay is going to protect our aquifer. But it's not everywhere, and it's actually not even always clayey. So it's not as protective as we had hoped. Okay, so moving below the surface and into your rock layers, this is Military Ridge, which I'm sure you're all familiar with, and these are the different geologic units here. So you have a whole bunch of these in green. That's called the Sinope group, and if we come over here, that's kind of like the topmost layer of your rock. You've got the Galena, that's actually what hosted a lot of the ore, and then you've got the Decorah and the Platteville. We're going to just lump them all together and call them Sinope. And you can see that north of Military Ridge, a lot of that is eroded off and not present. So we're going to see these underlying units. South of Military Ridge, we have a sort of a big, broad upland that's capped by that Galena dolomite. And our flow systems, that's going to sort of affect our flow systems. It also affects the topography. I mean, south of Military Ridge, it's a much broader, long plateau. North of Military Ridge, you got a lot more steeply incised valleys. That affects the land use. That also affects groundwater quality. So that's your sort of landscape. Underneath this Sinope dolomite, there is a shale layer called the Glenwood, and it's going to play an important role. Then you have the St. Peter sandstone sort of in purple here. A lot of people's wells are in the St. Peter. And then you've got another dolomite, the Prairie du Chien group, right? And then underneath that, you have a big, thick package of sandstones. So this is a much more complicated geology than we have in northeast Wisconsin. There, it's like all dolomite. Here you have limestone, shale, dolomite, sandstone, lots of different things. And that really, really affects your flow systems. Okay, before we can get to flow systems, we have to do just a little groundwater 101 here. So we're going to start with the water cycle, which I'm sure everyone knows because we live within it, right? But just to bring some terms up, oopsie, I keep hitting the wrong button instead of the pointer. When we have rainfall, precipitation, rain, snow, whatever, there's a couple possibilities for that water to go. It can do surface runoff and go to a lake or stream. It can evaporate from a lake or river. It can transpire through plants and go back up into the atmosphere that way. Or it can do the part that's important for groundwater, which is it can infiltrate into the ground. But just infiltrating doesn't make it be groundwater. Because if you think about it, like in the summer, when the crops are on the field and all the trees are cranking out the water, you're transpiring a lot of water. So a lot of rainfall comes in, and it transpires right back out to the atmosphere. So that water is never going to make it down to the groundwater system. The times of the year where water that infiltrates actually gets down into the groundwater system, Those are what we call groundwater recharge periods. And it's usually when the transpiration demand is pretty low. So think spring of the year before stuff leaks out. Think fall of the year when the vegetation dies back. And really importantly, think spring snowmelt, right? Because that's sort of when water is going to get past that root zone and make it into the groundwater system. Okay, so we're going to go through some terms here and then talk about groundwater flow. An aquifer, because you're going to hear that term a lot, is just any geologic unit that can store and transmit usable quantities of water to a well. Things like sand, sandstone, dolomites, those kind of units. The water table, you're going to hear about that tonight too. Basically, that's the top of the saturated zone. So right below the Earth's surface, we have what we call unsaturated zone. And in that area, like if you dig a hole in sand, it's like it doesn't fill up, right? You have air and you have soil moisture sort of clinging to the grains. Once you get below the water table, those pore spaces are totally full up with water. And that's why we call it saturated. It's saturated with water. Okay, so you got the unsaturated and the saturated zone. When we get to groundwater recharge, like I said, that's water that makes it past this root zone and into your groundwater system. Okay, so now that we got the terms down, let's think about how groundwater moves. A lot of people think that like groundwater flow is like weird and mysterious, but it's really not. Okay, like you all know, on the surface, water flows downhill. It does the same thing underground. Okay, except the thing that we're looking at is where is that water table? Right, so we've got areas where the water table is high, and there's a fancy term for the top to saturated zone. We call it the hydraulic head. Okay, so hydraulic head, water flows from higher hydraulic head to lower hydraulic head. So here's the high point. It's flown over to this stream. It's flown over to this marshy area. If you know the distribution of hydraulic head, you know which way the water moves. Right? And we figured that out by looking at things like well construction reports. I spend a lot of time looking at well construction reports. Okay, so then we can think about different parts of the landscape where the water is coming in here, and you have this water table high. That's where like a lot of water infiltrates, and then it moves over and it comes out here. We call those discharge areas. In recharge areas, you have generally downward groundwater flow, then it goes kind of horizontal, and then in discharge areas, it kind of comes up and out. So you've got that sort of focus to it. Okay, but you have a slightly trickier groundwater flow system. It isn't, let's go back to the simple one. Notice it's all the same stuff there, right? All you're looking at is flow lines. When you start mixing different rock layers in there, it gets trickier. So here we've got aquifers, but we also have things called confining beds, things like shales and clays, because water doesn't like to go through those. So you might have a flow system where the water comes in, goes out, and comes out of the stream, and if possible, it really would like to not, it doesn't like going through the confining bed and going down. It's difficult and takes a long time for it to do it. So you could kind of move in and move out without necessarily, I mean, some water has to go down there because it has water, but it's much more difficult for water to get to these deeper aquifers. And here we're looking at flow times of days to years. Here we're looking centuries to millennia. Right? So having a multi-aquifer system really complicates those groundwater flow patterns. Okay, so how does it work in the Southwest? There's a lot of short little flow paths because you have all those stream valleys, right, where the water just comes in at the ridge top and it goes out sideways at the nearby stream, right? So short little flow paths, ridge top to adjacent stream. Because you have different rock layers, and I'm sure many of you recognize this picture, this is Highway 151 near Dickeyville. I got the picture from Department of Transportation when it was first cut. But if you notice, like, here you got your Galena, your Decorah, your Platteville. Notice there's a line right here where all the seepage is. Right? It all comes down and it goes sideways. It's doing that on a shale layer. Right? So when you mix these different aquifers and confining units together, you get a lot of lateral movement of water where it comes down and it moves more sideways and only a little bit of it sort of infiltrates and goes through. So that's very characteristic of this area. And the other thing with all that lateral movement of water is you get a ton of springs. This is from our springs database, where you see kind of sparse springs all over Wisconsin. And then look at this just like clump of them down here in southwest Wisconsin, right? You have a lot of springs coming out on hillsides and hill slopes and things. Okay, Joel's going to talk about the upper and the lower aquifer. And so we need to sort of define what separates those. This upper aquifer is that Sinope group that I already talked about, right? So it's the Galena, the Decorah, and the Platteville. And then there's a shale layer here. It's only about 3 feet thick, so it doesn't actually show up as a separate layer in this diagram, but it's the Glenwood Shale. Underneath it, you've got the St. Peter sandstone, the Prairie du Chien dolomite, and then this big, thick package of Cambrian sands. There seems to be a real difference between this upper Sinope aquifer and these lower units. And a lot of times what you'll see when you look at the well construction reports, the water levels in this upper aquifer are often like 10s to 100 feet more than this aquifer down here. And we just talked about hydraulic head. Higher hydraulic head wants to go to lower hydraulic head. If that shale layer is in there, it's hard to do. If we connect them, it's really easy to do. And Ken's going to talk a lot about that. Okay, so just a little detail about the flow systems. This is down in Lafayette County at Platteville's Pioneer Farm. And I just wanted to show you, we do have lots more data that we're not going into great detail on about how we understand these flow systems. So a colleague who's now retired from Stevens Point, George Kraft, put these wells in about 15, 16 years ago. They were all 6-inch wells with little skinny 1-inch pipes in them. And each of those skinny pipes only has a 5-foot screen on it. So it's measuring hydraulic head over a very short distance. There's lots of them here. Each of these little dots is a well. Like, so there. We're going to look at this line as if we took a knife and sliced into the ground, and you could see the geology. So this is this line from B prime to B, right there. And what you've got is you've got the Galena, the Decorah, a shale layer, the Platteville, another shale layer, and then the St. Peter. And what you see is almost all of these psometers are up in that upper aquifer, right? And we're going to look at what the water level tell us about the flow system. We're going to look at the well on the top of the ridge, the well in the middle of the slope, the well at the toe slope, and then over here by the Upper Fever River. Okay, so top of the ridge, highest water levels, mid-slope, toe of slope, near stream. All the water levels decrease over time. These are continuous water levels measured from June of 2020 till the end of last year. So it makes sense. Top of the ridge, the water flows over to the stream in that upper Galena aquifer. But I also wanted to use this to illustrate the difference between the upper and the lower aquifers, and also how recharge works in this area. So we talked about recharge being when water gets in, right? Let's look at some of these big rain events. Here's a big rain event in September. All these water levels have a big pop up right there. I mean, not huge, 1/10 of a foot, right? Here's some in November. Here's some in November, right? So water levels suddenly go up like 2/10 of a foot. That's a recharge event. And typically it's happening within days of that rainfall event. So what it tells us is that recharge is really rapid. It gets into the system very, very, very quickly. And the big one is always right here in March with the spring snowmelt event. So that's what's happening in that upper aquifer. Now looking at the difference between the two, this is like a little diagram of the rock layers, right? So you've got all those dolomites. This is our top well up in the Sinope. There's our 2 shale layers, and there's our deep well down here in the St. Peter. This well is sitting here at about 915, 16 feet, and this one is sitting up here at about 1,012, 1,014 foot. There's 92 feet of head difference between those wells, and they're separate because of those shale layers. And that is what you get to know about the hydrogeology before Joel talks to you about water quality and the statistical factors. And if you have questions about any of that, you can ask at the end.
Well, good evening. My name is Joel Static. I'm going to present the study findings next. I wanted to start here with the 5 study objectives. These are the things we set out to accomplish for this study, and these are the things I'm going to go through tonight. Objective number 1 is to determine the extent of contamination. Number 2 is to identify fecal sources of contamination. 3 is to test for pathogens. 4 is to assess well and geologic risk factors that may be associated with contamination. And objective 5 is to assess land use risk factors that may be associated with contamination. And for each of these three objectives, I'll go through the approach, the key findings, and context and interpretation. I want to start here by showing how these 5 objectives fit together, particularly in terms of the wells that we sampled for the study. So starting with the circle on the left, there are approximately 16,000 private wells in these 3 counties. We randomly selected from those wells to complete Objective 1, determining the extent of contamination. Some percentage of those wells in Objective 1 were positive for bacteria or had high nitrate. We randomly selected from those wells, this time 138 wells, to complete Objectives 2 and 3, identifying the fecal sources and testing for pathogens. And then when we get to Objectives 4 and 5, we took that contamination data and we looked for associations with various risk factors. So I'll talk about those at the end. So we'll start with Objective 1, determining the extent of contamination. Our approach here was to randomly select from private wells across the 3 counties. The idea is that these 3 counties have similar geology and similar land use, and we wanted to get a representative estimate of the extent of contamination. The wells that we randomly selected were sampled in 2 synoptic events. A synoptic event you can think of as a snapshot because these samples were collected over a short period, just 2 days. So we collected samples over 2 days in November of 2018 and in April of 2019. Different wells were sampled in those 2 events, except some by random chance were selected and included in both of those events. Between those 2 events, we sent out over 3,000 solicitations to private well owners, and 25% of the people we contacted agreed to participate and submitted a sample. Overall, we collected 840 samples for this objective. These samples were tested for total coliforms, E. coli, and nitrate. The lab that did the analysis is at UW Stevens Point, the Water and Environmental Analysis Laboratory. They're very accustomed to doing these homeowner tests, so they have instructions and sampling kits they mailed out to the participants, and then the homeowners collected the samples on the specified days, brought them to us at drop-off points in the area, and then we drove them up to the laboratory the same day that they were collected. The 3 contaminants we tested for, I wanted to make a couple of notes about them before we move ahead. First of all, nitrate. Nitrate can originate in fecal sources like wastewater and manure, can also originate in chemical fertilizers, and low levels of nitrate can occur naturally. The groundwater quality standard for nitrate is 10 milligrams of nitrate nitrogen per liter, or 10 parts per million. Total coliforms and E. coli are related. These are bacteria. They're used to assess the sanitary quality of water. Total coliforms are more general. They can be found in fecal sources like manure and wastewater. They can also just live in the soil, so their non-fecal sources are possible as well. In contrast, E. coli, they live in the guts of animals and are shed in fecal material, so they're generally considered indicative of fecal contamination. The groundwater quality standard for both of these bacteria is 0 per 100 milliliters. All 3 of these contaminants are commonly used to assess private well water quality. That's the reason we use them for this objective. The tests are relatively inexpensive and routine, which means we could do a lot of tests in a short period of time, which is what we needed to do. The other thing is the data we produce from this objective, because these tests are common, is easy to compare to other studies or to future work. So let's go to the key findings for Objective 1. Overall, 42% of the wells in November and 27% of the wells in April were positive for total coliforms or had high nitrate. And when I say high nitrate, I mean greater than 10 parts per million. The table here shows the percentages for the individual contaminants. The top 2 rows in bold are the 2 events from our study. And then the bottom 3 rows are statewide percentages from various previous reports. I have these here for some comparison. So we see in November, we sampled 301 wells. 34% of them were positive for total coliforms. In April, we tested 539 wells. 16% of those were positive for total coliforms. Those percentages are similar or greater than the statewide percentages. For E. coli, 4 and 2% of the wells were positive in our study, not different from the statewide percentage. And then high nitrate, 16%, and 15% of the wells we sampled had nitrate greater than 10, which is greater than statewide percentages that range from 7 to 10%. Something worth pointing out about this objective and these results is We set out to get a representative sample. This figure here shows our 3 counties. Every dot on the map is one of the wells we sampled. So we did a good job. Our random selection did a good job of getting spatial coverage of the 3 counties. Those red dots are the wells that were positive for total coliforms or had high nitrate. Those are also spread throughout the 3 counties. The white dots are those that were negative. So for context for this objective, the first point here, I mentioned the comparison to the statewide averages. The second point I wanted to make is that groundwater contamination can change over time. This is something we knew ahead of time because factors like weather and the contaminant sources can change. So Dr. Muldoon talked about the importance of rainfall and snowmelt for groundwater recharge. That's what carries contaminants to groundwater. And of course, that varies over time. Contamination sources can change. For example, when and where manure is applied varies throughout the year. And for that reason, we use these synoptic events. So by collecting samples over a short period of time, we minimize the variation that would result from those kinds of changes. But we also wanted to capture contamination under 2 sets of conditions, which is why we had the 2 synoptic events. And my third point here is our goal was to get a representative sample, so we did that with our random selection by testing many wells across the entire region. And by using these common tests, this data can easily be compared to future monitoring or studies. Moving on to objective 2, identifying fecal sources of contamination. The fecal sources we were interested in for this objective were human fecal material. So we have septic systems in the area, about 16,000 across the 3 counties. There are also some fields that receive land application of septage or municipal sludge. It's a relatively minor source of human fecal material, estimated at about 1% of the volume of what's released from all those septic systems. We have then on the agricultural side, of course, pig manure and cattle manure. And the way we identify these fecal sources is through a technique called microbial source tracking. We're testing for microorganisms specific to these fecal sources. And this technique works on basic biology. There are some bacteria and viruses that only live in the guts of humans. They're only shed in human fecal material. And so when we detect it in a well, we know human fecal contamination was present. The same is true for pigs, and the same is true for cows. And this leads to an important point for objective 2. We're identifying fecal sources of contamination. There are other sources like chemical fertilizers. We're not going to detect them here. Our objective was about the fecal sources. Our approach here was to randomly select wells from those that had previously tested positive for total coliforms, E. coli, or high nitrate. The idea here was to increase our odds of sampling fecally contaminated wells. And that's because the tests we use, the sample collection, and the tests themselves are more laborious and more expensive. We simply can't do as many. So to improve our chances of successfully identifying fecal material, we selected from previously contaminated wells. So we sampled a total of 138 wells. It was 34 or 35 different wells in each of 4 seasonal events. We collected samples by 2 sampling techniques. One of them, we ran about 200 gallons of water through hemodialysis filters. That's what's shown in the picture. So that filter grabs the microorganisms as the water passes through. And the second sample collection technique is simply to fill a 1-liter bottle and bring it back to the laboratory. This table here shows the list of microorganisms we tested for, grouped by the fecal source. The thing I want to point out here, for each of our fecal sources, we tested for multiple microorganisms, again, increasing our ability to detect fecal contamination if it's present. On to the key findings for this objective. Overall, human wastewater and livestock manure contributed to private well contamination, and we saw that human wastewater was more common. The table I have here shows the number of wells positive for each of the fecal sources out of 138. Human wastewater, 66 wells were positive, cattle/ruminant manure, 33, and the pig manure, 13. And those numbers include 26 wells that were positive for multiple fecal sources. For context, the first point to make here is that there are few private well studies that do these fecal source tracking tests. Only about a half a dozen that have used these tests for private wells. So there's not a lot to compare to. And what we do have varies quite a bit. So these previous studies, the human wastewater or livestock manure, anywhere from 0% of the wells to 61% of the wells were positive for these markers. Excuse me. And then the study in Kewaunee County that Dr. Borkhardt mentioned, we also tested for these fecal markers, and we saw 25% of the wells were positive for human fecal contamination, and 34% were positive for bovine contamination. We didn't test for the pig manure in that study. So the percentages here compared to Kewaunee County compared to southwest Wisconsin, a little bit different in terms of the human versus bovine. There are lots of possible explanations there. The way that manure is managed, for example, in terms of the amount of land in grazing, or when manure is applied, or the area of land to which manure is applied, might differ between these two study areas. Same thing with the density of septic systems. Of course, southwest Wisconsin, a much bigger area than Kewaunee County. But also the geologies specifically are a little bit different. And of course, these studies took place at different times, so those factors related to weather could come into play here too. The second point here is about these fecal sources. We talked about the septic systems and manure. So septic systems, they're fixed in place, more or less continuously releasing effluent. So we think of these as a constant source of fecal material. Compare that to manure. Of course, when manure is applied or where the cattle are grazing can vary. So it's more of an intermittent source. So the ways, the when and where of these sources on the landscape may also have something to do with the fact that we saw more wells positive for human wastewater than for the livestock manure. And something I want to point out here with these fecal source tracking tests is we're identifying fecal sources. These results don't tell us where the coliforms or where the nitrate was coming from. And that's because we're identifying fecal sources of contamination, as I mentioned. Non-fecal sources of contamination can also contribute. It's also because multiple contamination sources can affect a single well, and that can include non-fecal sources or fecal material from other animals. And finally, these samples we collected for fecal source tracking were collected months after those original nitrate and coliform tests. So for those three reasons, these results in Objective 2, they do identify fecal sources, but they don't tell us where nitrate or coliforms were coming from. Moving on to Objective 3, testing for pathogens in these wells. The approach here, those same 138 wells we just got done discussing, we also tested those for pathogens. We used 19 genetic tests for viruses, bacteria, and protozoa. And something to point out here is some of these pathogens are specific to people, and what I mean is they can only infect people, but many of the pathogens we tested for are zoonotic. They can infect people or they can infect animals. So here's the key finding for this objective is that pathogen genes were detected in 66 out of 138 of those wells. This table lists the number of wells that were positive for each of those pathogens. I won't look at the specific pathogens, but we'll point out two things. Number one, we tested or detected a wide variety of pathogens, viruses, bacteria, and protozoa. And the second thing is, while we did detect some pathogens that only infect humans, those are the couple listed at the top of the table, many of the pathogens we detected could also infect animals, which means just this pathogen test by itself can't tell us the source where these pathogens are coming from. So the first piece of context here is about health risk. So we detected pathogens in drinking water, so that means there is some potential risk of illness. But the specific health risk depends on many factors, including what pathogen was present, how much was there, how much water someone would drink, whether or not there was treatment in the home, and the susceptibility of the individuals consuming the water. So that the specific risk will vary. And something to note here in my green box on the side, we use genetic tests to identify these pathogens because we're detecting pathogen genes. The tests don't distinguish between living and dead pathogens, but these tests are still very informative. They tell us, first of all, that pathogen contamination is possible. That means there's a pathogen source nearby, and that means those pathogens can move into wells. The second thing is these genetic tests correspond well with other types of tests, including those that do detect living pathogens. And then third, these genetic tests correspond to documented cases of illness. Studies have shown recorded illnesses being related to measurements with these same types of genetic tests. I mentioned a little bit here, my point number 2 about pathogen sources. All of these pathogens are fecal-borne, so there's some fecal source out there, but for a lot of them, because they could infect humans or people, just this pathogen test by itself doesn't necessarily tell us where they came from. And then third, we'll get a little comparison to other groundwater studies that tested for pathogens. So here I just have 4 other groundwater studies that tested for pathogens along with our data to provide some broad context. The thing to keep in mind as we look at these percentages is that these studies differ in what pathogens were tested, how many pathogens were tested, and how many times wells were tested. And all of those things will affect the percentage of wells positive. So for example, the first study on the bottom left across the United States and Canada, 15% of the wells in that study were positive for a pathogen. Those wells were typically sampled once or twice, but only for 2 or 3 pathogens, right, compared to the 19 genetic tests that we used in this study. Then moving on the top, in Kewaunee County, 24% of the private wells we tested there were positive for a pathogen. And there was a private wells tested in Canada. 45% of those were positive for pathogens. And then we have our results here in southwest Wisconsin, 48% of the wells. And then 70% of public wells in Minnesota were positive for a pathogen. That study tested for many pathogens, similar to the amount we tested here. And also those wells were tested 6 or 12 times each. And of course, the more times you sample a well, the more likely you are to detect a pathogen. Okay, let's move on to objectives 4 and 5. Assessing risk factors associated with contamination. The approach for objectives 4 and 5 was the same. The difference is what factors we looked at. So we started with the contamination data we just got done discussing for nitrate, coliforms, human wastewater, and livestock manure. And what we do is test for statistical associations between that contamination and various risk factors. And the risk factors were in the categories I have pictured here. So what I'm going to do is go through the risk factors that were important in each of these 6 categories and provide an example or 2 of each one to give you an idea of the key things we found or the themes that we saw for these 2 objectives. So I want to start by showing the objective 4 risk factors that we investigated. There were many. We had well characteristics. These came from well construction reports, things like the age of the well, the depth of the well, the depth of the well casing. We looked at well siting factors like the soil type, the land slope around the well. And then we considered geology. Dr. Muldoon described the geology. We had several geologic factors, including the bedrock depth, so how far or how shallow, I guess, the bedrock is where the well is, and the open interval geology. So that would be that part of the well that's actually drawing in water. What geologic unit is that open interval in? And then the third factor we considered was the topmost geologic unit. So that's whatever was at the surface of the geology where the well was, regardless of where the well was drawing water. And then finally, we considered groundwater levels and rainfall prior to sampling 2, 7, 14, and 21 days. So the first key finding from objective 4 is that well characteristics were important. And there were many of them that were important. I have listed here the well characteristics that were associated with one or more of the contaminants. And something to point out here is that these well characteristics, as you can imagine, some of them are related. For example, deeper wells tend to have deeper well casings, and we saw consistent relationships among those related well characteristics. So I'm going to show a couple of examples of the well characteristics that were important. So here's the first one. We're looking at the probability of total coliforms contamination increased with well age. We're going to look at a couple of plots like this. They'll be all set up the same. The risk factor will be along the horizontal axis. In this case, that's well age. So going from left to right, we're going from newer wells to older wells. And then the contamination is on the vertical axis. In this case, we're looking at the probability that the well is positive for total coliforms, going from 0% to 100%. And so we see here the association is that the older the well is, the higher the probability of total coliforms contamination. And this might reflect the fact that older wells are deteriorated and therefore more likely to be contaminated. It could also reflect changes in well construction practices over time, the materials used, or maybe it is that older wells tended to be drilled shallower, for example. The second well characteristic I want to look at is well depth, and here we see that the probability of high nitrate contamination increased with, I have it written as decreases with well depth. So shallower wells are more likely to be contaminated with high nitrate than deeper wells. So as you move along the horizontal axis from left to right, the wells are getting deeper, and we're looking at the probability that the well has high nitrate, greater than 10 parts per million. So the very shallowest wells we see have the highest probability of contamination, and the probability of contamination decreases as wells get deeper. And there's something I want to point out here using this as an example. This applies to all the risk factors, is that these associations are about probabilities. In other words, we see here shallower wells are more likely to be contaminated. That doesn't mean every shallow well will be contaminated, and it doesn't mean a deep well can never be contaminated, right? There's the probability aspect to keep in mind when we look at these associations. The next key finding or set of factors I want to look at is geology, and we saw that geology was important. All three of those factors I described were associated with one or more of the contaminants that we measured. The example I want to use here is for that open interval geology, and we saw that the probability of high nitrate was greater for wells that were in the upper aquifer. So I have a simplified cartoon version of what Dr. Muldoon showed with the two aquifers. All right, so we have the lighter portion on top is the upper aquifer, and the darker portion on the bottom is the lower aquifer. And then I've got my three wells, and that the dashed line there is the open interval. That's where the well is drawing its water. And so on the left, we have that shortest well, but the point is that it's open to that upper aquifer, and we see the probability of high nitrate for wells open to the upper aquifer is 26%. Moving to the next well, one that's open just to the lower aquifer, the probability of high nitrate is much lower, just 4%. And then interestingly, our third well cross-connects those two aquifers. That is, the open interval goes from the upper to the lower aquifer, and it's drawing water from both. And wells like that cross-connect also had an elevated probability of high nitrate. The next set of risk factors I want to talk about is rainfall and groundwater depth. These were also important. They were important for human wastewater contamination. So more rainfall prior to sampling, higher probability of contamination. And I have another cartoon here to show the idea of groundwater depth. We saw that wastewater contamination was more likely when groundwater depth is shallow. So we have our house on the left with a septic system releasing effluent, and we have a very shallow groundwater table, which means that effluent has a short travel distance to go before it hits the water table. In comparison, our house on the right, also a septic system releasing effluent. Now the groundwater is very deep. That effluent has a longer way to go before it reaches groundwater, and contamination is less likely when that's the case. And groundwater levels can change throughout the year. Now let's go on to objective 5 and talk about the land use risk factors that we investigated. We looked at these risk factors in two ways. One was the distance to the nearest, so the distance to the nearest cultivated field or nearest septic system. And then the other was the amount of that risk factor near the well. For septic systems, that amount was a count, and we did that count within 750 feet, 1,500 feet, and 3,000 feet of the well. For the fields, we did the acreage. So we see the picture, the example I have on the right, that circle, we have a blue dot in the middle with the circle. That's one of the radii that we considered. And so we would count the number of septic systems using geographic information systems, the number of septic systems within that circle, the acreage of cultivated land within that circle, and then we would look at the distance to the nearest septic system and the distance to the nearest field. Same for livestock farms and for those farms that receive, or the fields that receive sludge or septage. So the first key finding for this objective, we saw land use was important. Agricultural land use was associated with total coliforms, nitrate, and manure contamination. So the three factors that were related to contamination was the area of cultivated land nearby, the distance to the nearest cultivated land, and the distance to the nearest livestock farm. We'll take a look at one of these examples. So here for this plot, the example I have shows that the probability of high nitrate increased with the area of cultivated land near the well. This plot on the horizontal axis shows the acreage of cultivated land within 750 feet of a well. So a 750-foot circle around a well contains 40 acres. So what we're thinking about is the 40 that's immediately around the well. And the contamination I have shown here on the vertical axis is the probability that the well has high nitrate greater than 10 parts per million. And so we see in the bottom left of the plot, when there's no cultivated land near the well, there's about a 5% probability the well will have high nitrate. And as the area of cultivated land increases, the probability of high nitrate goes up too, until the maximum, close to all 40 acres are in cultivation, there's about a 55% probability the well will have high nitrate. To provide a little context, I wanted to bring back the statewide percentages I mentioned during Objective 1. The statewide percentages for high nitrate was 7 to 10%, so I put this dashed green line at 10% percentage, and we see when we compare that to our results for Southwest Wisconsin, that 10% mark occurs when wells in Southwest Wisconsin have 10 acres of cultivated land within that 750-foot radius of the well. So in other words, 10 out of 40 acres are in cultivation. We're at that 10% mark. So the 58%, there were 58% of the study wells had more than 10 acres. And so those wells all had a higher probability of high nitrate than the statewide percentage. The 42% of the wells that had less than 10 acres would then have a lower probability of high nitrate than the statewide percentage. My last example here is for septic systems. We saw that septic systems were associated with human wastewater contamination. That included the number of septic systems near the well, the distance to the nearest neighbor's septic system. Another key finding here was an association we didn't see, which is septic system factors were not associated with nitrate and total coliforms contamination. So here's our example of human wastewater contamination and septic systems. So this relationship shows that the probability of human wastewater contamination increases when there are more septic systems near the well. So the risk factor for this plot on the horizontal axis is a count. It's just the number of septic systems within 750 feet, going from 1 to 18. And then we're looking at the probability that a human wastewater marker is detected in these wells. And so we see the more septic systems within 750 feet, the higher the probability of contamination. Now for this human wastewater contamination, I don't have a statewide average because those data don't exist. So instead what we'll do is look at the extremes. On the left side, we see 44% probability of contamination when there's just one septic system around the well, right? And that one septic system is probably the septic system that belongs to the house where the well is, right? So it's reasonable but not definitive that this 44% probability of contamination has something to do with the septic system on the same parcel as the well. On the other side, we see at the maximum, when there are 18 septic systems within 750 feet of the well, there's a 79% probability of contamination. So if you have 18 septic systems in 40 acres, that's roughly 2 acres per septic system. So information like this might be useful if we're thinking about planning a subdivision or septic system density in a situation like that. So here's where I'm going to end, bring it back to the two general study goals that Dr. Borkart mentioned at the beginning of the evening. Is that number one, we wanted to get an assessment of private well contamination in the area. So we did that with objective 1, determining the extent of contamination, and with objective 3, we tested for these pathogens and identified many in private wells. And then we also wanted to provide findings that could help identify potential solutions. And we did that by identifying the fecal sources of contamination. We saw that both human wastewater and livestock manure contributed to contamination. And then finally, we just got done discussing the risk factors associated with contamination. And we saw that well factors, geologic factors, and land use were all important. So thank you very much for listening. I'm going to hand the microphone over to Dr. Ken Bradbury.
Thanks, everybody. You've been very patient. So I'm the last one here, so I'll try to wrap this up fairly quickly. I wanted to talk a little bit more about understanding your well and emphasizing some of these things that Joel just talked about. Because I'm a hydrogeologist and I think a lot about well construction. And just to remind you what we're talking about here, that wells, if you all have a well, the well has a casing, a steel casing. That's what you see at the surface. That casing rarely, if ever, goes to the bottom of the well. The bottom of the well is open. So when you see in this diagram, there's a open interval indicated by those dashed lines. That's where the water actually comes into the well. And then where water stands in the well, where if we drop the tape down to measure the water, that's probably representing the water table of the aquifer that the well is finished in. But as was shown before, deep and shallow wells can have different water levels, and that's kind of important here. So I want to explain that by going back to this diagram that Maureen showed of the different geologic units around here. And don't worry too much about the names here, but this is the diagram we showed before. And now I'm just going to cut off the left side of that diagram. It's the same diagram, just cutting off the left side here. And I've put a couple of wells there. And the wells, these are two wells with different casing. You see a deep well on the left, a shallow well on the right. And the dark lines are where the well is cased, that's where the casing is, and the dashed lines are where the well is open. And so you can see the shallow well is open near the surface in what's called the upper aquifer. The deep well is open deeper in what's called the lower aquifer. And in between, you can see a shale called the Glenwood Shale. That was in Maureen's diagram as well. I've just emphasized it a little bit here. And that shale is quite important, even though it's thin. In fact, sometimes when the well drillers are drilling, they don't even notice it because it can be just a foot or even just a few inches thick. But it's quite important in the geology out here in separating the upper and the lower hydrogeologic systems. So now I've added water. And so I've shaded these units. And so now you can see that the colors have changed a little bit, and we have a upper water table and a lower water table indicated by the little inverted triangles. And you can see the water levels are standing at different points in the shallow and the deep well. On the left, you can see the water level in the deep well is standing pretty far down even below where that Glenwood Shale is. On the right well, the water table is quite a lot nearer the surface. And we see this all the time when we go out, or well drillers go out and measure wells that maybe even on the same property, and the water levels are vastly different by perhaps 100 feet in two wells that are not as far apart as the distance across this room. And the reason is the wells are finished differently. Now, why do we care about that? Well, because water, as Maureen said, water moves downhill. It's going to move from higher to lower places. So the water wants to move down to this lower aquifer, but it can't because of the Glenwood Shale in the way. So if we have no contamination source, remember we need to have contamination, we have to have a source of the contamination. And Joel has talked about what all those sources might be, but both of these wells produce water. And so with no source present, I've got a little arrow there of water coming out of the well. And both these wells are producing good water. That, in this easy definition, that's no contamination, right? So no contamination, both these wells are producing good water. But now let's add a little contaminant. I don't care what it is, but we put something on the surface there and it starts leaking downward into the upper aquifer. And you can see now there's sort of a cloud of contamination in the upper aquifer. And the upper well, the shallow well, is now producing bad or contaminated water in some way. But the lower well, the deeper well, is still producing good water. Why is that? Even though the contamination is going around it, it's because that well has a casing that's going through and keeping that water in that upper aquifer from getting into it. And so and this is why two wells adjacent to each other, one might have contamination and one might not, because the wells are finished differently. Now, Joel talked about cross-connecting wells. So now I've taken, the diagram has changed a little bit, and that middle deep well, you can now see the casing is turned to dashed because this well is now open hole, no casing, from a very shallow point to its bottom. And the other well is the same. So now these wells are able to, that well is able to connect the lower and upper aquifers. And if we add contamination there, now we have a very bad situation where the contaminant from the upper aquifer can not only pollute the upper aquifer, but it can actually flow downward through that connecting well and get into the lower aquifer as well. And so now both wells are producing bad water. And not only that, but other wells that are finished in that lower aquifer may also be producing bad water just because of this one well. And why were wells drilled this way? Well, because in the past, people didn't always understand this, and a deeper, more connected well like this is likely to produce more water, but it's not necessarily the best water. And so when we look now at the results that Joel just shared, again, let's bring this back to the geology. First of all, here's the map that Maureen showed, showing the different units. And one of the interesting things to me, and Joel, I think, showed this, the first map with all the little dots on it, you can see that those dots were evenly distributed across the landscape, those places of contamination. So there were not clusters. And so one of the takeaways I've gotten from this is that this problem is widespread. It's not just a cluster that can be blamed on one thing or one land use somewhere. This is a broad issue that we have in southwest Wisconsin. And if we look at each individual situation, here are the nitrate results by aquifer. And they're done in the form of histograms here, where you have the frequency of a result along the vertical axis and the concentration of nitrate along the horizontal axis. So up at the upper left, this is the Sinope, or that uppermost aquifer, and it's clearly the most contaminated aquifer with respect to nitrate. It has an average, if you probably can't read from the back, but the average is 6.3 milligrams per liter, but the exceedance is 31%. 31% of the sample wells exceeded. In contrast, if we move straight down, the deep aquifer had an average of only 3.2 and an exceedance of only 5%. So quite a bit of difference here. We move to the right here, the connected aquifers, though, the connected wells, had an average of 4.7 and an exceedance of 16.8. So kind of much closer to just the upper aquifer. And we have a couple of other aquifers we haven't talked about, sand and gravel. We didn't talk much about that, but of course near the river valleys there are shallow wells that are finished in sand and gravel, and those were not sampled very often, so there were fewer sample numbers here, but there are some kind of intermediate contamination on those. Those tend to be rather vulnerable wells in many cases. And then we had a large number of the wells that we were not able to find construction reports for. Why not? Well, there's a lot of reasons. Some of them might be old and the reports have gotten lost over the years. Sometimes the drillers never turned one in, or it was misfiled or mislocated, or some other reason we could not find it. So there's a lot of wells that we do not know the construction of. What if you want to find out the well construction? This is where I think the homeowner should know what their well is constructed, how their well is constructed. Well, this on the right here is a example of a well construction report that a well driller will turn in. The forms have varied over the years, but this is one kind of typical one. The DNR has a website where you can find this for your own well or for wells in the area, and this is the address. I won't try to read it, but it's a pretty easy to use map-based website where you can pull up a map and you can draw a circle on it and try to find the wells and download your well construction report. So that's one source of information where people can find this information. Information on groundwater quality. UW Stevens Point has a great website where you can look at groundwater quality averages for any place in the state, and they're averaged by county, by township and range, or by section, depending if they have enough samples. And so that's and this is free to use, easy to get to, and the website is at the bottom of this slide. So that's something that we use at the survey all the time. Where to get help with your water? From the back, you probably can't read all this, but this is from the DNR's web page. They have a lot of different publications on how to deal with different contaminants in water, nitrate and bacteria, but also arsenic and manure in drinking water, flooded wells, many other contaminants. A lot of good information there from the DNR. So that's another place to go. Again, Stevens Point comes up here. Stevens Point, the Watershed Center for Watershed Science and Education produces a lot of good handouts about wells, such as this one, improving your private well quality or maintaining your home well water system, and they have others, and their website's right there too. So that's a great source of information. And then finally, when we talk about geology, I'll tout our Geological and Natural History Survey a bit. We put out a lot of geologic maps, water table maps, and so forth. We have a new map of the Quaternary geology of Grant County, a new map coming out on the bedrock geology of Grant County. We're working on new maps for Lafayette County right now. We have publications here from our publications page. Again, you can go to our website and find all this information. So there's a lot of information from homeowners who are trying to deal when you want to start dealing with these problems. And that's what I've got for you, Joe. So I think at this point we'll hand it back to you, and thanks for your attention.
So there were a couple of questions about sampling error. So this question relates to that. Any possibility of sampling error? Were the water samples taken by the homeowners? So maybe address that question of opportunities for errors in the sampling. Who took the samples? How were they sampled?
Okay, Joel?
Yeah, go ahead.
So for to answer that question, for the coliform test, E. coli and nitrate, those were homeowner collected, following the kits and the instructions provided by the Environmental Analysis Lab at UW Stevens Point. So that was homeowner collected. There's only a couple studies out there in the United States that have looked at sampling error when there's homeowner collected samples, and it turns out it's pretty small. So error is likely small. For the pathogens and the source tracking that Joel talked about, that was Joel and Aaron. So that's trained technicians using solid aseptic techniques for collecting those samples. Also for those samples, we have a variety of controls, very extensive quality control, quality assurance plan to ensure that when we get a result, we're very confident in it. So homeowner collected for the coliform and nitrate, technician collected for the microbial source tracking and the pathogens.
Yeah, and I'll just add to that, with a homeowner collected sample, coliform bacteria are everywhere, and so if you don't wash your hands well and you stick your finger in the bottle, you can create a problem. It's hard to change the nitrate or the E. coli, which is fecal matter, because you don't, hopefully, usually have fecal matter on your hands. Well, some people do, but usually not. So the nitrate numbers are pretty solid no matter who collects the sample.
Okay, related to that, someone wants to know if the statistical results are skewed by the fact that you are asking for voluntary participation, so you have willing homeowners and folks who are unwilling. Does the fact that you're only getting samples from those folks who are volunteering to take samples from their private wells, does that affect the results in any way statistically?
Yeah, so we randomly selected the wells, but yeah, as the question notes, we only got a sample from people who agreed to participate. So if there were differences in participation, I don't know what those factors would be, so it's potential but unknown. Anything you want to add? Yeah.
And so in terms of the frequencies, that is the contamination rate, that could be biased. So even though we strive for representativeness, you're right, there's always selection, what's called selection bias and that sort of thing. People that are willing to have their private well sampled. When it comes to the statistical analyses, there isn't an opportunity for that. That's because what we measured the independent variable, the risk factors. So we're not picking only those people that live near a field or only those people that have a lot of septic systems nearby. That ranges, and that's actually in the report. The ranges of those risk factors you will find in the appendix of the report. So there isn't really any opportunity there for bias. The bias could potentially lie in the contamination rates.
Okay, this next question relates to wellhead construction, and they're wondering, can wellhead construction contribute to insects and other things like earwigs getting into the well and causing elevated coliform?
So if you look at your well, there's usually a pipe that goes up that is the electrical lines that then go down to the pump. And there should be a tight rubber gasket that prevents bugs getting in there. And there's also usually a little screened area that allows connection to the atmosphere so you don't get like weird back pressure things and stuff. If those have degraded and bugs can crawl in, earwigs love moisture. And I have opened wells like to measure water level and stuff that were just crawling with earwigs. So it is really important to maintain your actual wellhead because yes, bugs can crawl in there and then that would affect your bacteria numbers.
I just want to add a point, and it's one that Joel alluded to, although I mean he had a lot of information to present. And it's given the nature of the last couple of questions. And that is that what we're talking about here, a study like this doesn't analyze an individual well. It relies on individual wells, those 816 homeowners, to provide information for the population at large. That's what we're assessing, not your specific well. So the example I like to use is my father, who's passed away for some time now, but this whole question about smoking and cancer. You know, I think we would all agree now that if you smoke cigarettes, you're probably more likely to have cancer. But my dad always liked to point out the 110-year-old who was smoking 2 packs a day and never had a problem, right? So we're talking about, an individual story doesn't dispute the group level findings. So if you have a well that's next to a farm field and it has low nitrate, that doesn't dispute the findings that overall, for the group of wells that we studied, that the closer you are to a farm field, the more likely you are to have a nitrate level greater than the health standard.
Okay, the next question is related to pathogens and which ones are most dangerous. So it asks, which of the 3 sources of fecal material carry pathogens that are most risky for human and animal health when water is consumed? And then I'm going to add a follow-up to that. What are some treatment options to remove fecal material from an existing well? I think.
Yeah, so all of the pathogens we tested for are fecal-borne. They can cause, we call acute gastrointestinal illness, so think vomiting and diarrhea. They vary in the severity. Some of that depends on the pathogen. Some of it depends on the individual person. In terms of which fecal sources, as I mentioned, most of the fecal sources, or most of the pathogens we tested could originate in both, so it's not necessarily that one is one fecal source is more or less risky in terms of people getting sick. There are also, by the way, some pathogens that were only pathogens of cattle or pigs. We didn't include that in the pathogen count, but those were included in the source tracking number. So those would be issues for cattle and pigs. You want to add anything, Mike?
How about treatment?
Oh, treatment, thank you. Yeah, you want to talk about treatment? Okay. Okay, so yeah, in terms of treatment, reverse osmosis is filtration. That's one thing that will get rid of the pathogens. Ultraviolet disinfection will take care of pathogens, and chlorination will take care of pathogens. Then, of course, boiling water will do the trick too, but not practical to implement on a regular basis necessarily.
Were there any other land use options included in any of the well circles? I think what they're looking for is other kinds of ways of thinking about land uses that you could relate to contamination. They give some examples like golf courses, industrial drainage, wooded areas, parks, horticulture production. Did you differentiate beyond just agriculture?
So I showed the list of specific land uses we use. So golf course, horticulture, we didn't use those. On the agricultural side, we would have really liked is to have information about when and where manure is applied and where manure lagoons are. We couldn't get those data. So what we used is this cultivated land layer that's from the U.S. Department of Agriculture that designates land as cultivated based on specific crop types grown in the fields over the past 5 years. So it includes things like corn and soybeans are typical crops. We included the fields that had permits for municipal sludge and septic applications, but not those other types of land uses, no. And that, by the way, that cultivated land layer from USDA does not include like hay fields. So that wasn't one of the things they designated as cultivated when they created that. I refer to it as a layer. It's a geographic information system layer that we used for this analysis.
Were any or were many abandoned wells identified in the study, and what is the risk to groundwater contamination of abandoned wells?
We don't really have a good record of when wells get abandoned. So at the state survey where Ken and I work, we have well construction records for the state that date back to 1937. Now, some of those wells, of course, are probably abandoned. I spent a lot of time with this study going through all 840 homes and trying to match a well construction report to them. A lot of times there'd be an old well, but I didn't know if it still existed or not. There's a lot where I could match using historic plat books and using property tax records and using septic records, or sometimes calling people and saying, hey, I have 5 possible wells on this quarter of a square mile. Is one of these yours? But none of it tells us whether we have an abandoned well. So we spent a lot of time trying to match well construction reports, but we don't have any information about abandoned wells.
How would you compare groundwater susceptibility of Northeast Wisconsin and Southwest Wisconsin? Would the additional groundwater protections being passed and being implemented over the Silurian dolomite be relevant and helpful here with some minor tweaks, or would we need to start from scratch?
I don't think the Northeast Wisconsin standards are appropriate in Southwest Wisconsin. It's a very different flow system. It's a very different type of karst. The type of agriculture is different. The way a manure is applied is different. I think the things that Ken talked about in terms of well construction are more appropriate than any kind of manure ordinances. That is my opinion, but I do not think that they would be all that helpful here, given the differences in the flow system. In Northeast Wisconsin, when you apply things at the ground surface, just given the way that aquifer works, being one big thick dolomite, and having Lake Michigan way down here and Green Bay way down here, when you put stuff on the land surface, it can penetrate hundreds of feet in that aquifer within a day or two. Okay? That doesn't happen here. It goes into the upper aquifer and it tends to move laterally. So there, what you did at the land surface affected this huge swath of where everyone had their well. Here, they're very compartmentalized.
So this is an opportunity for 2 scientists to argue in front of the public.
No problem.
That is, so what Maureen is saying is that, yes, well construction, unlike Northeast Wisconsin, unlike Kewaunee County, which strikingly, well construction, if you take a look at the published paper, well construction didn't matter at all. It was absolutely amazing that we didn't find those relationships. Whereas, Whereas here, well construction matters. But that's not to say, Maureen, that, that land use is unimportant here.
I think land use
So land use is also very, very important. And when we write the scientific paper and we take the next step in the next step of modeling, we'll be able to start to tease apart a little bit, assess what are the most important risk factors. We didn't rank them here, but we'll start to be able to get an idea of the impact.
There's also a difference in how we do cattle in both places. The manure load in Northeast is really high. It is a lot of liquid manure applied at high rates onto that landscape. Here it's a lot more rotational grazing. And so there's a real difference in the manure load too. So I'm not saying that some discussion of how manure is managed is inappropriate. I just think that you can't blanketly take Northeast regulations and put them down here.
No, I don't disagree. All I'm saying is what you do on your land matters, and the proximity and the volume of the contaminant source around the well really matters. I guess the good news here in Southwest Wisconsin is the phrase I used to use in Kewaunee County is that you can't construct your way out of the problem. Here, perhaps, if you had the money, you had the financial resources, you could construct a new well and perhaps reduce the problem for yourself. But on a regional level, land use is really, really important, both septic systems and livestock.
As someone who grew up in Northeast Wisconsin and spent most of their life in Northeast Wisconsin, I think you're both right. I agree 100% with Mo on the land use and the cattle and the amount of usage growing up in CAFO country like I did. I can 100% attest to what you're saying, but I can 100% agree with Mark too. So I just want to say you're both right, but I wanted to stand up too for Northeast Wisconsin that it's not the same, but it is the same in a sense.
We should go to a new question.
And I'm not picking on manure. All I'm saying is that land use is important. I think it's absolutely fascinating that the percentage of wells contaminated with bovine manure in Southwest Wisconsin is notably lower than what we saw in Northeast Wisconsin. Now, is that due to agricultural management practices here? Perhaps, I think Joel has the numbers on the percentage of farms that are on pasture as opposed to CAFO. That could be the case, but that's speculation. We didn't study that. But it's a reasonable hypothesis that it has to do with the farming management practices here as opposed to Northeast Wisconsin. And Joel, maybe you want to mention the grazing percentages if you know.
Yeah, sure. What Dr. Borkhardt is referring to, the NASS, the National Agriculture Statistics Service, details agricultural land use county by county. And in Kewaunee County, only 3% of the agricultural land is in grazing. In Southwest Wisconsin, it's 15%, so five times more. So that's one idea, I think, what Dr. Muldoon is referring to. Those grazing cattle are spread out as opposed to manure applications in Kewaunee County coming from manure lagoons and being spread. Yeah, but you know the other thing is when it comes to the contamination, is it land use or well construction? When we detect nitrate, when we detect fecal microbes, it's coming from somewhere, and that somewhere is the land use, right? So maybe well construction is more important, but there has to be a source for there to be contamination.
Nice lead to the next question. If a lower aquifer well is contaminated, can it ever produce good water again? How long might that take? And in this area, is there a recommended well depth?
Yeah. Yes, it could produce clean water again if the source is removed or changed in some way. How long would it take? Depends on where the well's located, the depth of the well, and what the aquifer is like right there. So it could take a few years, or it could take many, many years. I will say, as a analogy, many of you remember the Scholesberg mine that was closed back in the early 70s. there were a lot of wells there where they were contaminated with sulfate, not nitrate, but sulfate that was released as the mines were dewatered, and then when the mining stopped, the water table returned, and that released sulfate. So there were a lot of wells that were contaminated with sulfate, and once the water table recovered and the mines were reclaimed, eventually those wells were cleaned up naturally. So it can happen. We have some evidence that it does happen.
The well depth question.
Well depth question.
I can answer that.
Oh, yeah.
You can't give an absolute depth for how far wells could be cased because the synapse can be several hundred feet thick. Maybe you only have 50 feet of it, or maybe you have 300. Right? So you can't just say, oh, drill to 300 and you'll good, or drill to 250. You have to drill through the Glenwood Shale. But what's good about that is that we have tons and tons and tons of well construction reports. And as Ken mentioned, we're doing a bedrock geologic map of most of the counties in the area. So not only do we have what the surface geology is, we have what the layers are like with depth. And so you can say, okay, at this spot in this square mile, the Glenwood is 250 feet down or 150 feet down or whatever it is. So that information is available, but it's not like just a magic number.
This is a question about groundwater levels and water table levels. The question is the water table dropping in this area? What does tiling of fields to get rid of wet spots, draining of wetlands and sloughs have? What is the impact on the depth of the water table? And they note that they've noticed in the spring melt and spring melt is over in just a couple of days, and the creeks look like they would in August very soon in the spring. So their sense is that water table is dropping overall.
I think that I would say it's not, I don't think that is happening in the long term. We know that because we have some long-term monitoring wells that the USGS has run for many, many years. And I don't know, Mo, if you remember if there's a long-term trend there. I don't think there is. I think if anything, maybe up a little bit. Now, I will say that in this year and perhaps last year, things have been lower than usual. So when I say there's no, you have to distinguish, just like the weather, from long-term trends and short-term trends. This spring and last winter had been really actually abnormally dry, and so water levels now are low. Certainly, they're low around my house. A couple of years ago, they were almost at an all-time high. Back when we started this project, say 3 or 4 years ago, the water table levels were very high because, if you recall, we had a couple of extremely wet years in there. Since then, we've had some dry years. So if anything, I think the long-term trend is upward. Now, as far as draining individual field tiles, it certainly is possible that you could lower the water level in a field with tiling, and it would affect the field. It might even affect some wells around the field, but it's not going to affect the region. So, groundwater is kind of a local resource in that way, that what you do locally can have a local impact. Anything to add, Bill? Yeah, we see higher precipitation rates in recent years, and that seems to be making water levels higher generally.
Do deeper wells contain more heavy metals? Is the taste different?
It is possible that it's not necessarily deeper wells. Remember that we're in what's called the old lead zinc mining district, and they mined lead and zinc, and there were other metals that were in some of those deposits. If your well is near one of those mineralized zones, you may have some dissolved metals in the well, and those could affect taste. Taste is a kind of an individual thing, so some people seem to notice that sort of thing more than others. But I wanted to point out that the tests we've done here in this study are for just some simple things, bacteria, nitrate, and some pathogens, but we didn't test for metals and arsenic and lead and all those things. And so there are other things in the water that are not necessarily or really ever man-made or, result of society. They are naturally occurring. That doesn't mean that they're not bad for you. And so it is good if you think you're in an area where the water is tasting strange or that there was an old lead mine nearby, to get your well tested for some appropriate constituents there.
And some people really notice iron issues with their well in terms of taste. And the aquifers that are dolomite and limestone tend to, in general, have less iron. Several of the sandstones, I noticed we lumped them all together, called the deep aquifer. Some are more iron-rich sandstones than others. And so if your well is drawing water from one of the more iron-rich sandstones, you have the potential to have it taste more irony.
Let's see, are there plans to do similar tests in other regions of the state?
I'm retired.
Not at this time, I guess is the answer to that.
So I think, in seriousness, Joe, in seriousness, as I said in my little 10-minute spiel, I was reluctant to work here because I was neck deep with Kewaunee County stuff. But Madeline was right, and I think you said it too, Mo. This hydrogeologically was fascinating. It's really interesting. And so it is a scientific advance, what we're going to be publishing. In other parts of the state that are hydrogeologically different, like where Linda is now residing, yes, it'd be worth doing these things. And maybe eventually we'll get the entire state. How many hydrogeologic regions are there, Ken? Maybe we get it all covered. So I think scientifically it is worth doing.
Does your study show or conclude that in Grant County, septic systems are the main cause of water pollution? Is it greater than other sources of contamination?
So in terms of the fecal contamination, we did detect more wells with human wastewater. I've got an appendix in the report that shows the contamination results for each county. I grouped everything together for the presentation and for the main part of the report. So you can look at that appendix to see county by county what the results were. But so we saw overall there was more human wastewater contamination in terms of fecal contaminants compared to the livestock manure. But keep in mind, when we looked at our land use associations, the nitrate and coliforms were associated with agricultural land use, but not with septic system. So when we ask what's the biggest cause of contamination, what contaminant are we talking about? If we're talking about wastewater, it's the septic systems. We saw those associations. If we're talking about nitrate and coliforms, those were associated with the agricultural land use rather than septic systems.
The other thing to understand is that a lot of the human waste, human fecal material that we detected was detecting a microorganism that is one of the friendly bugs in our human gut tracts that help us digest our food. So it wasn't pathogenic. But what we can do and will do in southwest Wisconsin is similar to what we did in Kewaunee County, is that we know the source, and in that same sample we can also see the pathogen. And so in Kewaunee County, that second paper that was on the screen takes a look at the health risk. We have a way of evaluating health risk from the drinking water of the private wells. And in that study, even though the human wastewater was about the same as the livestock, they were about equal in the contamination, the pathogens were more common in those wells with bovine manure, in which case most of the illness, what we conclude in that paper, and has been, surprise, surprise, controversial in this state, most of the illness came from bovine manure. We can do the same thing here in southwest Wisconsin.
A related question, if human wastewater or septic is not responsible for nitrate or total coliform, or not associated with nitrate and total coliform, then what contamination does it produce? So in other words, what is the issue with septic wastewater?
Good. Okay, so the first point here is septic systems can release nitrate. They are a potential source of nitrate and total coliforms. Just at a broad scale, we didn't see the associations with septic systems. They do, I mean, it's fecal contamination, so we're talking about whatever goes down the drain in people's houses, it gets flushed down the toilet. That's the contamination we're talking about. We're most concerned generally when that contamination includes pathogens, which of course sick people shed pathogens in their fecal material, and so that's one of the concerns with human wastewater contamination. But as Dr. Borkart mentioned, the microorganism we detected most frequently was a friendly bug. So in one sense, if you can get over the notion of what's in the water, that one won't hurt you, right? But if the pathogens are coming along too, which is possible, then that can be an issue.
I was just going to add that when you have septic system impacts to groundwater, we specifically were looking at pathogens. UW-Stevens Point does an analysis specifically to look at septic system indicators, and there are other things in terms of trace amounts of pharmaceuticals and artificial sweeteners and things like that that you may not want to be drinking as well. So you can tell the wastewater sources apart by looking at the pathogens, but there are other things in septic systems that you may not want to be passing to your neighbor.
And Joel is putting it too politely. There's just a high probability that you're drinking your own poop with a private well. Yeah, well, I think it was if you have the one septic system, there's 44% probability of having wastewater. And there's nothing wrong with that. There's nothing wrong with that at all. It's just that if someone visits and then they deposit a pathogen into your septic system and you drink that, well, then you've got a problem. So in general, the human fecal waste and the pathogens that are in that waste are not good. You don't want to pick one of those up. So it may be, like I said, in Kewaunee County, most of the pathogens were associated, even though it was equal contamination rates, most of the pathogens were associated with bovine. We haven't looked at the data that way here yet, so we don't know. But in general, all of us drink a little bit of fecal material all the time, and it doesn't hurt us. It's only when that pathogen's present.
Go ahead.
I'd just like to reiterate something Mark said before, between the general and the specific. You know, from the tone of the question, it sounds like someone's concluding that, oh, septic systems aren't a problem, and that's on the general average, maybe not, but that doesn't mean the septic system next to a well can't contaminate the well. That's definitely possible. For nitrate, yeah, for nitrate. So anyway, just remember the conclusions here are the general conclusions. We aren't looking at any specific well or any specific septic system.
And there was a question about manure pits. Was that something that you looked at? There was a concern about manure pits potentially contaminating, especially if there are any karst features. Was that something that you looked at? If not, why not?
Yeah, good. That's something we wanted to look at. And in fact, that in Kewaunee County, the data were available for manure lagoon locations. So that was a risk factor we looked at. Turns out it was important for contamination in Kewaunee County. We did not have those data for Southwest Wisconsin. I think Lafayette County had a layer of manure lagoons. The other two counties had partial layers. We tried looking at air photos to identify these manure lagoons to create our own database of where they were, but we couldn't do it with high enough confidence. So we didn't. The bottom line is we didn't feel good enough about the data we have to try to do these tests. So we wouldn't have been confident in those results. So we used the cultivated land and the livestock farms instead for agricultural land use. Oh, go ahead.
You also had a question about karst and whether we looked at karst features. We don't have maps of karst features for these three counties. So we couldn't look at proximity of a karst feature to each well and whether it contributed. Creating a map of karst features is very time intensive, and it was well beyond the scope of this study to map the karst features in these three counties. So it was also not included. It may very well be important, but we couldn't look at it because we didn't have the data set and we didn't have the resources to create the data set.
If you want to have your well water tested, the page the handout gives you some options. Also, each of the county extension offices in Grant, Lafayette, and Iowa County have test kits. that will, if you take your sample and with that test kit, you will send it to the UW Stevens Point lab that was involved in this study. Those kits have all the information about how to take the sample. It is $60 for the homeowner package that will do total coliforms, nitrates, and several other tests for you. There are other places that you can get your water tested as well. County health departments typically do it, but look at that information sheet for more ideas on where you can get your well water tested. Anything I'm missing from any of our presenters or land conservation folks that you want to share with the audience before we wrap up?
I guess for Grant County, on our county webpage, we have a link to, right on the conservation sanitation zoning page, that we have a link on the top directly to the Iowa County extension page that does give all the SWIG information, so you can get that through the Grant County page.
Anybody else? Join me in thanking our presenters and researchers. We cannot overemphasize the value that their work is providing to this region, to the state, to the nation, really, in terms of understanding groundwater quality, and drinking water quality. So thank you very much to all of you, to our land conservation offices. They're the ones who are responsible for helping you do a better job of taking care of the land and the water resources. So thanks to them as well. Thanks for coming out tonight. I guess we'll be hanging around if you have questions, but thank you again for coming.